Hello everyone, I'm going to present the fastest way to do the bulk RNAseq or in a microarray or in a chip sequence data analysis using the NIH Geo to R tool. So for that we have the beautiful Google Chrome and we go from there. Let's go to the NIH Geo. And you can see geo means gene expression omnibus, where people submit their data. So we go to the geo and start searching in a specific disease or conditions which you'd like to look for. For example, on this content, we can search something like acute myeloid leukemia, probably. A lot of data set came. So I selected human. And then we selected expression profiling by high throughput sequencing. One of the major things to notice in here is that not all the data sets have this facility like J2R because it's a new facility and NIH has certain conditions those need to be followed before submitting the data, and only then this J2R facility will be available on those specific data set. Let's go. So when we are going through this, we can see there are a few data sets where we have J2R analyze J2R facility. So let's go. And we will probably select one of them. This looks nice, a lot of data set. Let's select one of them. For example, we have this data set named as mitophagy promotes resistance to BH3 mimetics in acute myeloid leukemia. It was published by RNAseq from NYU Grassman School of Medicine. Let's say pretty much recent, you can say. You can visit the paper. There's a publication. This looks promising. Let's try to figure out their RNAseq data. RNAseq, site-seq, a lot of data. Data ability, bulk RNAseq is submitted. This is bulk RNAseq data, so we had interest in there. Let's go. This is our data set. RNA sequencing was performed to explore the molecular mechanisms underpinning the MCL1 inhibitor resistance. Expression profiling by high throughput sequencing, and we have the number of samples we have are 7. 2 of them are controlled probably, and 5 of them are treated. Let's go. So this is the data sets we have. So first thing we will do is go in here. I looked at this data set earlier. That's interesting. And we divide them within multiple groups. So first, we take the parental cells without treatment group as a control. So we select control and we select the treatment group. Select these 2, assign them to the control. Select these 4, assign them to the treatment. And you can see control and treatment group. Now we just click the analyze, and analyze this start. You can take your stopwatch and it will probably take a few seconds. So what happening? It's analyzing the data. So the data is processing, and in the meantime they have this within 5 seconds very beautiful visualization figure, which used to take hours after hours for the people to learn the basics of programming and do this analysis. So the first question that comes in our mind in here is that what was the algorithm and the programming language at the very first was used to analyze this data set? Let's go to the script where you can specifically see the entire algorithm that was used. So they have used the R version 4.22 as Babel's jQuery and Lima. And for this analysis specifically they have used R-based DESeq 2. This entire script, if you're interested, probably you are not using this facility. So either way, if you have interest, later on let me know. I will do the analysis. I'll do explanation in alphabet by alphabet or letter by letter if you are interested. These are some of the parameters those used in here. So we have many figures in here, and I will explain each of those figures one by one. A very short introduction. Let's first go to the dispersion estimate graph. What does it say? It's mean of normalized count, which we did using the DESeq algorithm, and dispersion of the genes. So if you look at this specific figure, what you see, the gene estimation has the fitted figure and the final results. So this is a trend line, and if the mean of normalized count is low, where we expect the dispersion more, and when the normalized count is higher, we expect a trendy line, the fitted line. And the beautiful part of it is that this data looks good. It looks good. And when you did the control versus treated and you went through the genes in here, the DESeq, which has used the negative binomial modeling to generate this entire analysis, looks well fitted in here. Based on the genes and their expressions. So the next thing that we'll be looking is this p-value histogram. The frequencies of p-adjusted values. And if you look at in here, and the p-values are adjusted based on false discovery rate, which we have in here. You can see the false discovery rate we have done based on the Benjamini and Hodgeberg false discovery rate, which really is good for this analysis. Actually, I can explain some other day. So if you look at in here, what you see, that the p-value, which is the more reduced, the differential expression genes is more concentrated in this area. When the p-value increase, the genes on this area probably doesn't have differential expression value as expected. So we will be mostly concentrating on these values when we are looking at the data. And afterwards, next part is to look at these box plot. It is also known as box and whisker plot of normalized count, because we have this y-axis locked in normalized count, and we have each of the samples. One of the major thing to concentrate and look at in here is that the median line of the gene expression in normalized count. So this is a kind of a median line, and we can compare sample to sample, and we can compare sample group versus the treatment group. So in the control group, the gene expression and treatment group gene expression is little bit different, which is probably expected. And this central tendency probably gave us this central tendency of expression of genes, give us a better view on sample to sample versus control versus treatment genes normalized count. Then we start looking at our data in a different aspect, and that is based on the gene expression. We built this UMAP, which is Uniform Manifold Approximation and Projection graph, and this is a 2D visualization of high-dimensional gene expression. Here we expect the control group gene expression will be distinct than the treatment group, which is clearly visible in here. This is the treatment group, and this is our control group, and this clearly denotes there are differential expression of the genes across the control and treatment group, and they are distinct based on control and treatment group. So this is important. We extensively use the UMAP and TSD plot in single cell analysis. It could be single cell DNA, single cell RNA, single cell site C, which we have proteomics part also, and we have single cell DNA plus protein. We can use data. I'm not sure much of the applications. Yeah, we do apply in there, but it's a different perspective to bring in here. So if you have interest on it, this analysis, just let me know. I can just text me or send me email, comment out, or whatever the way you want to approach. You'll find the way if you want to communicate. So the next part of our analysis is to look at or identify the genes those differentially expressed. So when you do the control versus treatment comparison, you see almost all genes are down-regulated and up-regulated, which is not true. So based on these, we need to do some optimization in the analysis pipeline. So we get back in here and we set up the log fold accordingly. So we have this figure. We see, okay, probably most of the genes are like these, so we select. Let's select the log fold change. Okay, 8. We do the reanalysis. This is the beauty of it. It takes few seconds, and you can reanalyze this entire data based on the evolving optimized threshold you want to set in there. So the analysis will be mostly concentrating right now on the genes those are probably is really distinguished expressed and differentially expressed. Let's look at this. These genes are down-regulated, these genes are up-regulated. Now the question comes. We have in x-axis we have log fold change, in y-axis we have log 10 value, p-adjusted value. So the genes in positive side 0 to 15, that is the genes those have differentially expressed and highly upregulated, and the genes in negative side is downregulated. Control versus treatment is written in here, but it's very tricky and very interesting to see that how the NIH has defined this control versus treatment. There could be different ways of defining this control versus treatment. One could be is that you take the control genes and then you go to the treatment group and see after the treatment in comparison of control genes what are the genes those were upregulated or downregulated after the treatment. There could be another way of identifying that in comparison of treatment, we look at the control and then we look at try to figure out that what are the genes those are upregulated and downregulated. It entirely depends how you write the script. So if you go to the script and there's a portion, you see the groups make names in here, control versus treatment. See control treatment, where you define this specifically what you want to compare against whom. I would suggest to very very carefully look at these, because it could figure out that comparison was wrong. I found few cases where the result was reversed. So I would say, if you're interested, let me know. I will show the cases in there. It's just the wrong position of the control versus treatment, where which one would be. Now let's go back in here. We want to explore and download. You can explore and download this list. These are the top differential expressions and tell list they have given. You can look at the symbol and you can look at the p-adjusted values, and then you can look at the log-fold change values. So let's see. If we want to see, okay, this is the list we have. We want to look at these. Okay, top upregulated. This is PRDX, or we want to look at it here. CFC. Text 15. There are lots of genes which is in here. You can see, and you can just click on this download list and you can get it then downloaded and you can explore them. So the option is available in there. This is probably the most important figure for your analysis, and it's something that you will be presenting. The next part is looking at this mean difference plot, which some people do, the MA part. The idea in here is to say the mean in x-axis locked in mean of normalized count and look at the log to fold change in here. You can explore and you can also download this list. MPO in here. Then upregulated, downregulated genes. So it depends on your interest, which one you want to look at. Either you want the volcano plot, where you have log to fold versus p-adjusted value, or you want the MA plot, mean difference plot, where you have log normalized, mean of log 10 mean of normalized count versus log to fold change, and you can use them based on your experiment interest, and you can download the list and explore the genes. Now finally we have this Venn diagram. Here the Venn diagram is saying that when you use the RNAseq algorithm and try to figure out the genes based on the p-adjusted value less than 0.05, out of 19,169 we found 819 genes which are differentially expressed genes, including the upregulated and downregulated. Here also you have the freedom to just select it and download the significant genes from here. So essentially this is the entire analysis you have. You don't need to do much. Just click in there and get it done in a few seconds. Let me know if you have any question or any concern. Thank you.